Yes, yes, it's
That's good.
good time yet.
Cheers. Will you be talking in Spanish?
A little bit at the beginning, and then I will give the introduction in English, and then you follow. Speaking in Spanish. A little bit at the beginning, and then I have a little introduction in English, and then the introduction to each of you in English also.
So
Yes. ¿Comenzamos? 2 minutos. So, if I well understood, you went to Cuernavaca today, or
No,
you are going
I
to
will
be
go
tomorrow
tomorrow.
at the Genomic Science Institute.
Yes, and Dr. Esperanza Martinez will be my host.
And what time is your talk?
I think it's at noon. I know I will get picked up at 9:45.
Okay.
And the talk will be a little... Yes. Yes? Somebody's talking to me. I'm the guinea pig here. Yeah.
Bueno, vamos a comenzar. Let's begin. Buenas tardes. Sean todos bienvenidos al segundo día de actividades del simposio El mundo del RNA, pasado y presente, organizado por el Colegio Nacional y magistralmente coordinado por el doctor Francisco Bolívar Zapata, el doctor Eusebio Juaristi y Cosío y por el doctor Antonio Lascano, miembros todos ellos de este Colegio Nacional. Esta tarde escucharemos 2 conferencias, la primera de ellas a cargo del profesor Ulrich Müller, adscrito a la Universidad de California en San Diego y especialista en ribosimas, estos pequeños fragmentos de ácido ribonucleico con actividad catalítica. Y la segunda conferencia estará a cargo de la profesora Marie-Christine Morel, académica de la Universidad Pierre et Marie Curie, así como del Museo de Historia Natural en París, Francia. La profesora Morel es especialista también en ribosimas, las cuales estudia en condiciones fisicoquímicas extremas, así como también en el tema de viroides, un tipo de virus subcelular que puede proporcionar información sobre formas de vida antiguas. En atención a nuestros conferencistas, me voy a permitir compartirles una breve introducción a las temáticas que se tratarán este día en idioma inglés. So it is my pleasure to welcome you to this Rna World Symposium. And as a way of introduction, I would comment the following. To fully understand the processes occurring in present-day cells of all living organisms, it is necessary to consider how they arose in evolution. Among the most fundamental problems associated to this understanding, it is the expression of hereditary information, which today requires an extraordinary complex machinery that proceeds from DNA to protein through an RNA intermediate. That calls our attention. How does this machinery arise? A possible explanation comes from the hypothesis of an RNA world, proposed to have existed on Earth before modern cells arose, and which places the RNA as a molecule with the ability to store, transmit, and duplicate genetic information by itself. RNA is believed to have once been capable of supporting independent life forms. It was previously thought that the only biomolecules that could catalyze essential chemical reactions in cells were proteins. However, the discovery of ribosomes, a class of RNAs capable of catalyzing chemical reactions, considerably strengthens the Rna World hypothesis. Later in evolutionary time, DNA took over the role as the guardian of the genetic material, and proteins became the major catalyst and structural components of cells. However, the transition out of the RNA world was never complete, as RNA still catalyzes several fundamental reactions in modern-day cells, a role that can be viewed as a molecular fossil of an earlier world. Nowadays, opinions differ as to whether RNA constituted the first autonomous self-replicating system or was a derivative of an still earlier system. The recent findings that activated pyrimidine ribonucleotides can be synthesized under plausible prebiotic conditions indicate that it is premature to dismiss the RNA first scenarios. Additional evidence supporting the concept of an RNA world results from research on viroids. Viroids are mostly pathogens with a few hundred nucleobase genomes, highly complementary, circular, single-stranded, and non-coding RNA fragments. They are proposed as living relics of an RNA world and have attained potential significance by representing the most plausible macromolecules known, capable of explaining crucial intermediate steps in the evolution of life from inanimate matter. Today's conference will be dissected by two experts in some of the previously mentioned aspects of the ribonucleic acid molecules. The first lecture corresponds to the participation of Professor Ulrich Müller from the Chemistry and Biochemistry Department of the University of California in San Diego. Professor Müller, or Uli, as known by his friends, did his undergraduate studies in chemistry at the LMU in Munich, Germany. Then continued his PhD in biochemistry at the Darmstadt Institute of Technology in Germany also, and did postdoctoral studies with Professor David Bartel at the Whitehead Institute in Cambridge, Massachusetts. This is where Uli learned to work with ribozymes and in vitro selection. The research interests of Professor Müller's laboratory are centered in the catalytic RNA molecules, the ribozymes, as well as in the group 1 intro ribozymes and their evolution in cells. Professor Müller has a prolific list of publications in high-impact journals of his area of expertise. He has been responsible for projects economically supported by NASA, the National Science Foundation, and the National Institutes of Health in the United States. He has supervised the research activities of several PhD graduate and undergraduate students. The title of his lecture is How to Generate RNA Polymers in an RNA World. So please, Professor Müller.
Thank you. Is this switched on? Yes. So first, thank you very much for inviting me to this very nice conference. I want to show you the results on our research regarding how RNA could have been generated in an RNA world. And let me start with the very general picture, how the question, how did life begin? And you know that the crust of the Earth solidified about 4.5 billion years ago. And the next thing we know is about 3.5 billion years ago, we find evidence for single-celled organisms that look like life. And as you know, these single-celled organisms then diversified into eukarya, archaea, bacteria, and into today's living organisms. What we know very little about is about this time between 4.5 billion years ago and 3.5 billion years ago. And this is where I modified my slides since yesterday, because I wanted to incorporate what Krishnamurti said and what Mike Yarrow said, and put my slides, my presentation into context with those presentations yesterday. You remember that Krishnamurti talked about how the first RNAs and DNAs could have been generated and selected. Mike Yarrow talked about a first possible minimal genetic system that could have existed during that time. And I will be talking about the next step about ribozymes. Ribozyme is the name for catalytic RNAs, and how they could have functioned in a mature RNA world. So I will be talking about the step afterwards. And the picture you will be getting from my talk will be one of a pure RNA world. And I do not deny that the RNA world started in a very messy environment. It was, I agree with what Krishnamurti said and Mike Yarrow said, we started from a messy environment, and lots of these chemicals that were around during that time were important for the rise of the RNA world. So let's first look at what constraints we have to think about the origin of life. And the reason why we need to think in this way is because the origin of life study is a very peculiar field of research. You may have noticed that in every area of biochemistry, for example, you have the object of study right in front of you. And you can address the question, how does that work? In our case, our object of study died out 3.5 billion years ago. So we cannot study it directly. The only question we can address is, how could it have worked 3.5 billion years ago? So that's why we need to approach it a little bit on the indirect side. First of all, I want to give you a definition of life. This is my definition of life, and I know some people have slightly different definitions of life. Life is a self-replicating molecular system that is capable of open-ended Darwinian evolution. So the evolution is a central part in the definition of life. A second criterion is the second law of thermodynamics. As you know, this is one criterion, one constraint that we cannot escape. Every form of matter has to observe the second law of thermodynamics. And when you look at what happens in self-replication, you are generating a lot of order, a lot of structure. According to the second law of thermodynamics, this would be impossible if it would not be coupled to another process where you increase this order dramatically. So here you have a process where you form biological structures, where you have a positive delta G, which means a decrease in disorder, that needs to be coupled to another process. And you can also see it from the perspective. This is an energy-consuming process. This is an energy-generating process. This is a very lax definition. Now, as Krishnamurti explained to you yesterday, it is very important to look at the chemistry that may have existed during that time. And he also mentioned, for example, the Strecker synthesis of amino acids. And it's important to note that the chemicals that existed during that time on Earth may have been very different from the chemicals that exist today on Earth. And the last point, the last constraint that we need to think about, how did life originate, is that by definition, these life forms evolved stepwise into today's life forms. So there must have been some continuity in the mechanism of information transfer, basically the copying of the genomes, and in the metabolism. And we will come to this point in a second, because as it was shown or discussed yesterday, I think 3 times, and I'm discussing it a 4th time now, the RNA world hypothesis was formed by Crick, Rose, and Orgel, and I think I need to add Rich Roberts too, because they wanted to solve the chicken and egg problem. Which one came first? DNA, which today encodes genomic information, or proteins, which do most of the catalysis. So these two are dependent on each other. And these people realized it is astronomically unlikely that a system in which these macromolecules, protein, are dependent on the other macromolecules, DNA. So they speculated that RNA, in addition to its known role and known ability to encode genetic information, it could also catalyze chemical reactions and possibly mediate its self-replication. And as you may also know, in 1982, Tom Cech and Sid Altman found that yes, RNA can indeed catalyze chemical reactions. Now, this is very conceptual, and it's trying to address the chicken and egg problem, which one came first. Since then, there has been quite a number of evidence supporting this RNA world hypothesis that said, yes, there was indeed a stage in the early evolution of life in which RNA had very important functions, more central than today. And one of them is the ribosome. The ribosome is a catalytic RNA. Everything you see here in blue is RNA. Everything you see in white are the proteins that are on the periphery of the ribosome. What you see here in the middle are 3 tRNAs, and this little squiggle down there is a messenger RNA. The star is where the catalysis happens, and the star is not close to any of these ribosomal proteins. We know by now that RNA is what catalyzes the peptidyl transfer in the center. Since the protein synthesis is made by a catalytic RNA, that means that this form of encoded protein synthesis must have come after catalytic RNAs were there. And similarly, when you look at how DNA is synthesized in today's organisms, you see that the 2' hydroxyl group is removed from an RNA nucleoside triphosphate to give you a DNA 2' deoxynucleoside triphosphate. Similarly, the C5 methyl group is added to an RNA deoxynucleotide to give you a DNA deoxynucleoside triphosphate. And this makes most sense in the context of an RNA world that's being taken over by DNA. So these are just 2 pieces of molecular fossils that support the idea that yes, there was an RNA world in the early evolution of life. Now, since I'm a chemist / biochemist, I want to understand how this worked on the molecular level. And as Ram Krishnamurti talked to us about yesterday, was the generation of nucleosides and perhaps RNA polymers from very small prebiotically plausible molecules. The steps that I'm most interested in is the step of chemical activation and polymerization. Let me quickly say about this here. As Ram Krishnamurti described yesterday, this is an area of intense research. How could nucleosides have been generated in an RNA world or before an RNA world? And I'm not an organic chemist, and I leave this to the organic chemists to figure out how could this have worked. I just assume that by definition, if there was an RNA world, there must have been a prebiotic way to generate nucleosides. And I start from there. The first step is you need to take nucleosides and add a phosphate, a 5' phosphate. Because again, then you get these nucleotides or nucleoside 5' phosphates that are the building blocks for RNA. The second thing is, we talked about the second law of thermodynamics, that you need an energy source to drive the polymerization of RNA in an aqueous system. So if you are dissolved in water, it is thermodynamically unfavorable to polymerize nucleotides into RNA polymers. And this energy source can be supplied by this molecule here, cyclic trimetaphosphate. I will go into this molecule on the next 2 slides in detail. For now, I just want to say this reaction between the nucleoside 5' hydroxyl group and the trimetaphosphate could have led to nucleoside triphosphates, and that can then be used to polymerize into RNA polymers. First, I want to convince you that Trimetaphosphate is indeed a prebiotically plausible molecule. And there are 3 different mechanisms known how trimetaphosphate could have been generated in a prebiotic world. The first of them was discovered in 1991, where you take calcium phosphate, basalt, and you heat it up in volcanic environments to more than 1200 degrees Celsius. And then you generate polyphosphates, including trimetaphosphate. The yield of this process is not very high, so I'm not a big fan of this process. This process here uses acidic ammonium dihydrogen phosphate and heats it up in the presence of urea. Then you only need a temperature of about 100 degrees and you generate polyphosphates. Now, we may find the lower temperature of 100 degrees very interesting, but to have high concentrations of urea and acidic ammonium dihydrogen phosphate may make it less prebiotically plausible. The most interesting way of generating trimetaphosphate is that from meteorites. And that may strike you as a little strange, but keep in mind that meteorites have been raining down on the early Earth in massive amounts. And for example, between 10 and 50% of all the phosphorus inventory of the Earth comes from these meteorites. So in early times, there was a large supply of these iron-nickel phosphides, or schreibersite, and that slowly eroded in the presence of water. So you can erode these over hundreds of years, and once the phosphide is in contact with water, it slowly oxidizes to phosphites, and these phosphites can then be further oxidized to polyphosphates. Now, the interesting thing about this process here is this intermediate, this phosphite or phosphonate, has been found in 3.5 billion year old marine sediments by Matt Persek in his 2013 paper. And it was found as a precipitate with calcium, which shows that the concentration of this intermediate, this phosphite, was at least 0.1 millimolar in the prebiotic ocean, which is a lot, at least in areas of the prebiotic ocean. So this is my favorite process to generate trimetaphosphate. And we have, which is very rare, direct evidence that the intermediates of this process existed. All of these processes, they generate not just one molecule, they generate a spectrum of molecules: phosphate, pyrophosphate, trimetaphosphate, linear triphosphate, linear tetraphosphate, and so on. And you see that the yields of these ones are, of trimetaphosphate, is in the range of 0 to 10%. So under some conditions, you could have generated quite some trimetaphosphate. Now, if you want to think about, would this have been a good starting material for an Rna world organism, a good food source, you need to think not only about how could it have been generated, but also how stable is it. And this is a recent result that I got yesterday from a student by email. So that's Sam Borowski in the lab of Judy Kim, who has a collaboration with us. She looked at the reaction of trimetaphosphate with water that generates linear triphosphate. So this is the hydrolysis and the inactivation of trimetaphosphate. When you take a Raman spectrum of trimetaphosphate, you find that at about 1160 wave numbers, there is a peak that's very specific for trimetaphosphate, because this is such a nice symmetric molecule. So you can follow the decay of this trimetaphosphate over time. This is on day 0, after 9 days, after 48 days, and after 151 days. You see the signal here disappearing, and this disappears relatively fast because this is done at pH 1.9. We did this experiment at multiple different pH values, 1.9, 2.1, 3.1, and 3.9. And you see that the decay kinetics are fastest at a very low pH, and they get slower and slower the closer you get to a neutral pH. Now, when you look at the lifetimes here, at the decay times, you see why our experiments at pH 7 didn't give us any useful results yet. Because this trimetaphosphate is stable for years at neutral pH, at least when we extrapolate from the lifetime in days, which is coming from these curves here, and you plot against the pH values. Other people have found that there is a linear relationship in this range. So if you extrapolate these values to a pH of 6.5, which I think is currently the best estimate for the pH of the Archean Ocean, then you get a lifetime of about 2,200 days, which is about 6 years. I have to admit that these experiments, this is just our first set of experiments, was done without magnesium. And in the presence of magnesium, this lifetime will probably go down, but I assume that is still in the range of years. Altogether, these results suggest that trimetaphosphate had a lifetime in the range of years in the Archean Ocean and probably was able to accumulate, at least in some parts of the Archean Ocean. For example, in tide pools where you can have evaporation. So this was to convince you that trimetaphosphate could have been a plausible energy source for an Rna world organism. Next, I want to show you the reaction that we want to use it for. So this is an experiment that was done by Leslie Orgel in 1978, where he incubated 20 millimolar adenosine with 200 millimolar trimetaphosphate, and he found that in 1 hour at room temperature, he had about 5% of these nucleosides triphosphorylated at the 5' end. Now, you see a problem here. This reaction only works if the pH is in the range of 12. At the pH of 12, RNA is not stable. So first of all, we repeated this experiment and found, yes, at pH 12, it works nicely. And we also found that, yes, at pH 7, nothing is happening. So our goal was, can we find catalytic RNAs that can catalyze the triphosphorylation of these hydroxyl group at neutral pH? And now I'm coming to the first big part of my talk. This is the in vitro selection of catalytic RNAs or ribozymes that catalyze the triphosphorylation of RNA 5' hydroxyl groups. And what I'm doing here is two things. First of all, I'm going through the concept of in vitro selection, and I'm trying to make that clear for everybody. And the second thing I'm doing is I'm trying to go into the specifics, why we were setting up this process of in vitro selection exactly like this for the specific case. So we start with a pool of DNA molecule. This is double-stranded DNA that contains a number of 150 randomized positions. So imagine 150 random positions. At each position, you have the possibility of 4 nucleotides. So if you have 1 randomized nucleotides, you have 4 possibilities. If you have 2 nucleotides, you have 64 possibilities. With 2 nucleotides gives you 16 possibilities. 3 nucleotides gives you 64 possibilities. So 150 nucleotides gives you 4 to the 150 possibilities. That's about 10 to the 100. And keep in mind that the Earth has only about 10 to the 50 atoms. So we would never, ever be able to cover the whole sequence space of a sequence with 150 random nucleotides. However, it was found that for these in vitro selections, using a tiny fraction of that pool, of that complexities, is enough to obtain catalytic RNAs. So now let's look at the next step. Here we have this pool molecule with a promoter for T7 RNA polymerase encoding a hammerhead ribozyme, so that after you transcribe this DNA molecule into an RNA molecule, you have this hammerhead ribozyme at the 5' end and this random sequence with 150 nucleotides at the 3' end. The nice thing about this hammerhead ribozyme is it cleaves itself off very efficiently during transcription, so that you end up with a 5' hydroxyl group and a randomized sequence of 150 nucleotides. This is important because this 5' hydroxyl group is exactly the reactive group we want to have reacting with trimetaphosphate. So in the next step, we are incubating this RNA molecule with trimetaphosphate in the presence of magnesium, trimetaphosphate, and a buffer. those molecules that can fold into their right three-dimensional structure to catalyze this reaction, they will decorate their own 5' end with a triphosphate. So this here is the reaction that we are looking for. We would like to find molecules that can do that. Now, the second most important step in this in vitro selection cycle is how do we fish out these molecules that have triphosphorylated their 5' terminus. To do this, we used a ligase ribozyme. That's the R3C ligase ribozyme developed by Jerry Joyce. This ligase ribozyme and a short biotinylated RNA. You see here that these pool molecules get annealed with this ligase ribozyme and this biotinylated RNA, such that if there is a 5' triphosphate, this ligase ribozyme will catalyze the covalent link between the 3' hydroxyl group and the 5' end of this pool molecule, and then you have a covalent connection between 150 nucleotides of the pool and the biotin at the 5' end. Which means most of you may have worked already with biotin, or you know that biotin can be used as a very high affinity and high selective tag to pull out molecules from a mixture that is very messy. And that's exactly what we were doing here. We were using biotin, this biotin tag, to pull out these pool molecules, wash all the other pool molecules away, so that now we have selected these active molecules. Now keep in mind that this may just be 1 or a dozen molecules, which is a number of molecules you cannot work with. So now we're using reverse transcription and PCR amplification to amplify that selected pool to amounts you can work with again. Now we are back here at a double-stranded RNA pool, and this pool is now enriched for those sequences that can catalyze the reaction. Now let's think about the numbers a little bit. We start with 1.7 times 10 to the 14 sequences. And let's say it could be that there's just one molecule in the original pool that's catalytically active. How many cycles of this in vitro selection do you think are necessary until the pool is dominated by active sequences? Keep in mind that in every biochemical experiment, you have a little bit of carryover. And we found out that the enrichment of the active sequences in one cycle is 10,000-fold, 10 to the 4-fold. So if in our example we have one active sequence in 10 to the 4 molecules, after one cycle, we have 1 in 10 to the 10 molecules. After 2 cycles, we have 1 in 10 to the 6 molecules. After 3 cycles, we have 1 in 10 to the 2 molecules. And after 4 cycles, our pool should be dominated by active sequences. And the beautiful thing of this in vitro selection was, this is indeed how it worked out. After 4 cycles of in vitro selection, our pool was dominated by active sequences, and we made 4 more rounds of in vitro evolution to enrich for the most active sequences. And here I'm showing you results from the first 36 isolated sequences, isolated clones. So Giannina Moretti, who was doing this in vitro selection in my lab, she randomly picked 36 clones and tested them for their activity. And we saw that at least 34 of these 36 showed catalytic activity, which is very good for an in vitro selection. It basically means we had close to no false positives. The other thing you see here is these are the sequences aligned, these 36 sequences. And you see that 24 of those 36 are single sequences. Only the rest has multiple clones, which means you can expect from these numbers that we probably have on the order of 100 different sequences. So we were very encouraged by this result. And in a collaboration with Irene Chen at UC Santa Barbara and A. Pressman, Irene Chen did a high-throughput sequencing analysis of these in vitro selected pools. And what she found was we have more than 300 completely different clusters that were all highly enriched over the rounds of selection. So here you had the round 4, round 5, round 6, round 7, and round 8. In round 8, you see these red sequences. The red sequences become present in the final round 8 pool. And the blue sequences arise from a mutation in one of them and make them more active so that they have a higher frequency in the final pool. Looking at this enrichment behavior over the rounds, Irene Chen was able to predict which ribozymes are expected to be most active. And she predicted these 8 sequences here to be very active. And in this plot, they are shown together with those 36 sequences that Janina isolated previously. And you see that we have a 10 to 15-fold higher activity of the best ribozymes than what Janina found before, which shows you the power of high-throughput sequencing analysis. You can really find the best active clones by that. Another nice outcome of this study was she was getting a large view on hundreds of different ribozymes, and she was able to make predictions computationally how active they are, depending on how they enriched. And one thing she could then obtain from that is when she estimated the activity, how good catalysts these ribozymes were, and how frequent they were in the pool. She found this curve in red is the curve that best described the behavior. So this means that there are lots of sequences in the pool that have a low activity of about 0.01 per minute. There are fewer and fewer sequences and very few sequences that are more active than about 0.1 per minute. So this shows you something about the analysis of the activity of clones in the pool. And we are following up on this study to get a more detailed view on how you can use high-throughput sequencing to analyze pools from in vitro selections. Now, I want to go back to the biochemistry because in the end, you need to test biochemically how active are these clones. And we developed an assay to directly look at the triphosphorylation reaction. The first thing we did is we radioactively labeled these ribozymes, these catalytic RNAs, throughout the whole sequence. And what we intended to do was to react these ribozymes with trimetaphosphate such that some that have a 5' triphosphate can now be discriminated from those with a 5' hydroxyl group. And we wanted to do this with a gel shift experiment. But of course, if you have a 182 nucleotide long RNA, you cannot discriminate in a gel shift whether this RNA has a 5' hydroxyl or a 5' triphosphate group. So what we did was the following. We used a catalytic DNA, a DNazyme that was developed by Jerry Joyce, to cleave off 8 nucleotides at the 5' end of these ribozymes. Now you have short 8 nucleotide long RNA molecules that are radioactively labeled and that do or do not have a 5' triphosphate. In addition, you have these other ribozyme fragments. What you see here on the right is an analysis using this assay, and that is showing these large ribozyme fragments up there. This is a denaturing polyacrylamide gels in which the short fragments run down here. And here you see 2 controls. This is one control where we had 100% of a 5' hydroxyl group, and this is one control where we had 100% of a 5' triphosphate. Now you see over increasingly longer times of incubation with trimetaphosphate, we find that the signal for the 5' hydroxyl group disappears, and the signal for the 5' triphosphate increases over time. And then we plotted these time courses, the activity, for 8 different ribozymes that were analyzed in this way. And one nice thing is all of them gave perfectly single exponential fit. So the behavior was very relatively easy to understand. Now, we wanted to choose one of these ribozymes for a more detailed analysis. And because we wanted to have something that forms to a large extent into one catalytically active structure, we took this ribozyme here that appears to react to an amplitude of about 83%. This is the ribozyme I will be discussing you on the next few slides with you. First of all, we reduced this ribozyme in length from 182 nucleotides to 96 nucleotides. And we did a secondary structure probing analysis with this reagent 1M7. This reagent was found by Kevin Weeks, was developed by Kevin Weeks for secondary structure analysis. We may not want to go into how the secondary structure analysis works, only that those regions that are flexible are accessible to these probes, and then you can annotate on your sequence which nucleotides are flexible and try to find secondary structure that are consistent with that. So this was the only secondary structure we found that was consistent with the secondary structure probing data. In addition, we did some base covariation analysis in which we tested, for example, whether this CG base pair was formed. When we took the CG base pair and measured the activity, we had a rate of about 0.03 per minute. When we mutated this C to a G, such that we would have a GG pair, which of course doesn't form a Watson-Crick pair, activity dropped to 0. When we mutated this G to a C, such that we had a CC pair, activity dropped to a very low level 2. But when we made both mutations, there was a GC pair, so base pairing was restored and activity also came back up. Those types of experiments are very useful to test whether you have a specific secondary structures forming. They not always work very nicely. So in this case, for this other pair, you see that, yes, activity drops and it comes back a tiny little bit. And in those cases, very often not only the presence of a base pair is important, but also the identity of a specific base. So this is the current best secondary structure, or the best secondary structure we had at that point. And then we took this ribozyme, and Greg Dolan in my lab, he did something called a doped selection, which is basically the same in vitro selection that I described to you before. But it is exploring the sequence space around the most active ribozyme. It is not covering a very wide sequence space. It is exploring variants of this ribozyme. And what he found was 7 mutations in the sequence that increased the activity by 24-fold to 0.31 per minute. And for those of you who have worked in the lab, this is about as fast as you can pipette. So Greg was working really fast for these early time points. So now that we had this evolved ribozyme, it was also becoming apparent that probably there's another secondary structure element forming there. And that was confirmed in later experiments. And so that the new secondary structure looks like this. This 3' tail bends around and makes this additional stem. What Arvind Kupi did in my lab, he fragmented this ribozyme into 3 RNA strands, and then he truncated this stem off, which was completely dispensable. So that now you have a very small ribozyme that consists of an RNA strand of 14 nucleotides, of 19 nucleotides, and of 13 nucleotides. And this is in the range when you can start thinking this might be a plausible size range, what could have originated in an origin of life scenario. So I want to summarize the results so far of these triphosphorylation ribozymes that use magnesium to catalyze the triphosphorylation. First of all, and the most important point is, these results show that yes, catalytic RNAs could have used trimetaphosphate as an energy source for RNA world organisms. The second thing we found is we found hundreds of ribozymes in this pool of 10 to the 14, more than 300, which suggests that this is not a very rare activity. Other ribozymes seem to be much harder to find. So this is also good news for an RNA world organism. It may not be that hard to evolve this activity. And as Arvind showed here, is these ribozymes can assemble from multiple short RNAs, and that's also good news for an RNA world organism because you don't need to make 96 nucleotide long RNAs. You can get away with 34 nucleotides or shorter. What we have not yet found are ribozymes that can triphosphorylate 3 nucleosides. We have tested the existing, 8 of the existing ribozymes with 3 nucleosides, and they were not active. And if I see you maybe in a year, I may be able to tell you more about our current experiments on these efforts. Now, I want to add one more short story to this triphosphorylation ribozyme story. It's just a one-slide story. And this has to do with switching the magnesium 2+ ions for lanthanides. And you may think, that is crazy. Why would he use lanthanides? And what got me interested in that was lanthanides are known in this 1995 paper to catalyze the hydrolysis of trimetaphosphate by the lanthanide coordinating those 3 negatively charged oxygens on the trimetaphosphate. And the important thing is that the ligands of these lanthanides have a strong effect on how well this catalysis goes. Here you see the catalysis in the presence of NTA as a ligand. Here you see the catalysis in the presence of EDTA as a ligand. And you see that nitrilotriacetate was by far the best ligand to promote this catalysis. Now, when you think about catalytic RNAs, a large number, perhaps the large majority of them, uses metal ions at their catalytic center. And you can see these ligands here as just placeholders for RNA. So RNA may be able to coordinate those lanthanides. And depending on how the RNA coordinates these lanthanides, it may be able to catalyze the reaction to very high levels. So this is just telling you chemically it's plausible that lanthanides should be able to be catalyzed to catalyze the nucleophilic attack of RNA 5' hydroxyl group on this trimetaphosphate. Because hydrolysis is nothing else but the nucleophilic attack of water on this trimetaphosphate. And the other argument I want to show you here, although lanthanides belong to the rare earth metals, they are not that rare as their name suggests. This here is the abundance of lanthanide in the Earth's crust in ppb. And it shows you here, these are the rock-forming elements like silicon, oxygen, magnesium, that are quite frequent with a frequency of about 10 to the 4 to 10 to the 8 parts per billion. Then you see lanthanides have a frequency of about 10 to the 1 to 10 to the 4 in frequency. So they are not far behind those rock-forming elements. Only with those rare metals like gold, iridium, osmium, then you have really rare elements. So gold, iridium, they should be labeled the rare earth elements. Anyhow, the point I want to make is lanthanides are not that rare, and they actually got concentrated highly during the solidification of the Earth's crust. So from that perspective, lanthanides may not be that bad. Now, what we did is an in vitro selection, or actually Kevin Sweeney in my lab did this in vitro selection, and he just replaced magnesium 2+ in the incubation with ytterbium 3+, one of the lanthanides. And I save you the whole process of the in vitro selection. I just show you the results. Here is the assay that he had about a week ago, where he showed out of his 34 clones, I think, altogether, most of these clones were either weakly active or inactive. One single clone was very active. And the other thing you see with this single clone, you have quite a lot of degradation there. So what I think is happening, and we will find that out over the next weeks, is lanthanides are such good Lewis acids that they not only could help the catalysis of the triphosphorylation, they are such good Lewis acids that they degrade the RNA too. And of course, if you are a ribozyme, there would be 2 ways how to use those lanthanides. One way would be to stay away from them. Don't get too close to them and bind them very weakly. And then you may have a weakly active ribozyme. And the other possibility would be to bind them strongly and make a fast reaction. And then you not only see a strong signal, but you also see degradation bands. So this is an intermediate result that I'm showing you here. But my feeling is that lanthanides were too hot to handle for ribozymes. They are such good Lewis acids that if they are coordinated by RNA, they may degrade the RNA. So this is the end of the first part of my talk. And the second part, so the first part was about this chemical activation. How can you go from a 5' hydroxyl group to a chemically activated 5' phosphate? And the second part will not be on the chemical activation. It will be on the polymerization of nucleoside triphosphates. And you may have seen this ribozyme before here. This is the ribozyme that was developed by David Partell in 2001. And this is one of the hallmark ribozymes in the origin of life field, because this ribozyme is able to bind a primer and template and use nucleoside triphosphates to extend the 3' end of the primer in the template-dependent fashion. So here you see one of these polymerization experiments where after, so you have time points of 0 hours, half an hour, 3 hours, and 24 hours. After 24 hours, you can see an extension of about 14 nucleotides. Now the nice thing is, this is not only showing that polymerization works, David Bartell also cut out these bands and sequenced them and showed that the fidelity of this ribozyme is quite good. It's in the range of 97%. And that's close to what you would need for a self-replicating RNA organism. Now, the structure of this ribozyme is known only partially. It's known of this catalytic domain here. Of this accessory domain, we do not know the structure. So we very nicely know how the nucleoside triphosphate is bound, how it is chemically activated, and how it is positioned at the 3' end of the primer, such that you can extend the 3' end. What we do not know is how this accessory domain works. And the secondary structure I'm showing you here was developed by Peter Unruh, where he showed that there is a close contact between these nucleotides here, and this domain seems to fold on the top or on the side of this catalytic domain. And what he suggested is that this purine-rich loop was responsible for binding nucleoside triphosphates. But it was a speculation at that point. And we wanted to see, can we use an evolution experiment to answer that question, whether this is indeed the nucleotide binding, the NTP binding site. So what we did was the following. To set up this evolution experiment, it's always good to tag your molecules that are successful with some chemical moiety, and then you can select those successful molecules based on pulling on this chemical moiety. So in this case, we connected first the 5' end of the ribozyme to this template here, so it was solidly, nicely connected to the ribozyme. And the second thing we did is we connected the 3' end of the ribozyme to the 5' end of the primer. So now what happens is, if we extend this 3' end of the primer, the ribozyme is actually extending its own 3' end. And if we extend the 3' end of the primer with, for example, 6-thiol-GTP, 6-thiol-GTP has a sulfur in there, and that adds a tag to the ribozyme, a sulfur tag. Additionally, 6-thiol-GTP is a nucleoside triphosphate that the ribozyme has not seen before. It hasn't been evolved in this context. So you could expect that if this ribozyme is forced to get very efficient in using this 6-thioguanosine triphosphate, then it makes some adjustments in its sequence, and especially in those parts of the sequence that bind the nucleoside triphosphate. So to select those molecules that have a sulfur modification, we use mercury gels or APM gels. So those are polyacrylamide gels where you generate an intermediate phase that has mercury covalently immobilized, such that when you run these RNA molecules on this gel, If you don't have a thiomodification, you just move through this thiomodified part of the gel. If you do have a thiomodification, you get stuck right on this interface on the top. The next thing is you isolate your ribozymes from this little sliver of the gel. You reverse transcribe them, and which means you transfer the RNA into DNA. And after doing that, you can PCR amplify them and introduce mutations, which means now you introduce mutations all over this ribozyme. And in the next round of selection, you're selecting those variants that work the best. And this is called an evolution experiment because we are not starting with a huge pool of sequences and all we do is select. This is what we did previously. In this case, we are starting with just a single variance and we are doing mutagenesis, selection, amplification. Mutagenesis, selection, amplification. This is exactly how natural evolution works. And by doing this, you successively accumulate those mutations that are the most beneficial. Here you see how this experiment worked. We used specific concentrations of the 6-thio-GTP. We changed the reaction times in response to the pool becoming more active. And we always measured what fraction of the pool is getting stuck at this interface. And from converting these data to an estimated observed rate, you see that this initial pool was getting about 2 orders of magnitude more active than the initial ribozyme. Arvin Akopi, he was the student who was doing this experiment. He isolated 27 sequences and he tested them all for their catalytic activity of this ligation of 6-thio-GTP. And here, this is the list of these clones. What you see here is the list of 41 mutations where we found a mutation in these ribozymes. And what you see is there's one mutation at position 156 that exists in every single of these clones that went through these 10 cycles of evolution. Additionally, we, for example, have 3 of the best sequences carry this mutation at position 79, and a number of the best sequences carry this mutation at position 113. So what Arvin did is he took this most efficient clone 28 and he analyzed which of the mutations in clone 28 is necessary for full activity. And he found these 6 mutations were not all necessary. For example, the mutation at position 24 can be removed without any loss in activity. The mutations at position 146 and 187 can be removed and you even increase activity, which means these 2 mutations were deleterious for activity. In contrast, there are 2 mutations that have a weak effect on activity, position 79 and position 113, and the one mutation at position 156, which had about a 50-fold effect of activity. So you can already guess from here, these 3 mutations are the one that mediate activity, but we haven't shown that. In this experiment, we have only shown these 3 mutations are necessary. To test whether these 3 mutations are also sufficient, he generated the wild-type mutation, the variant with 3 mutations, and then all intermediates with 1 mutation, 2 mutations, and the 3 mutations, and measured their activity. And now you can see how these mutations behave in the ribozyme. You see the mutation 118 from wild type to 118 does not give you an enhancement activity, which means this mutation by itself is not beneficial to the ribozyme. You saw that this mutation 156 had probably a large effect, and here we have about a 50-fold increase in activity, which is consistent with the results we saw before. The mutation at position 79 had about a 2-fold increase in activity. Now the interesting thing is when you now look at the combination between mutation at 156 and 113, you see now that this activity increases from here to here. Although originally 113 was not beneficial, that shows that 113 and 156 are 2 cooperative mutations. the beneficial effect of 113 is only seen in the context of 156. And the mutation at 79 always seems to have about a twofold effect of improving activity, which suggests position 79. This mutation acts independently from the other mutations. And now we can have a look at this polymerase ribozyme and try to get some structural understanding. How may this ribozyme bind nucleoside triphosphates? This here is the crystal structure of the catalytic core of the ribozyme. This is the position where GTP is bound. The mutation at position 113 is here. The mutation at position 79 is here. And we found in these kinetic assays that 113 and the mutation at 156 were acting cooperatively. And because of this large effect here, we think that 156 and 113, they are the two mutations that really improve the binding of the nucleotide. And that confirms the hypothesis by Peter Unruh that this purine-rich loop here is the major determinant in binding the nucleoside triphosphate in this polymerase ribozyme. So with that, I have shown you our results on chemical activation using trimetaphosphate and on how the polymerase ribozyme binds nucleoside triphosphates. And I want to show you the people who did the work. Arvin Akupi, Kevin Sweeney, Greg Dolan, and Janina Moretti. Janina Moretti did this major paper for us in which she established these in vitro selection for triphosphorylation ribozymes. Judy Kim has a student, Sam Borowski, who did the resonance Raman spectra for us. And funding came from NASA and NIH. And before I end, I want to go to one last slide that is not on the translator's copy, I think. This is a Google image of the UC San Diego campus of Urey Hall. And here was Stanley Miller's office in Bonner Hall. And there is a beautiful little tree down there and that I always liked. And I was imagining that Stanley Miller, you know Stanley Miller, the father of origins of life chemistry, that he was sitting here under this tree, under these benches, and was thinking about origins of life chemistry. And one day, people, the UCSD crew, they cut off a branch of this tree, and I was able to get a little piece of wood from that, and I manufactured it in a little figure. And this is a little thank you to Antonio for inviting me to this wonderful conference. So this is from a little figure from what I call the Stanley Miller tree at UCSD. Thank you for your attention. I'm sure we have some questions.
Thank you very much, Uli, for this splendid lecture. Tenemos tiempo para preguntas, por favor. Las preguntas las pueden formular en español y nos ayudan con la traducción, o en inglés, como se sientan cómodos, por favor.
It seems clear to me that ribosomes can activate nucleotides. But thinking in a further step, is it possible that the activated nucleotides can be polymerized only by catalytic RNAs? Because trimetaphosphates are very stable molecules, as you said, at pH 7. So how can these catalytic RNAs may achieve a breakdown of a phosphoanhydride bond between two phosphates?
I think this is a question that addresses a problem in many areas of the early steps of an RNA world. So the question was, how could it be that trimetaphosphate may have been used in an RNA world, but could it have been used before? Do you really need an RNA to catalyze this? Is that right? And so I think the whole field is waiting for some miracle catalysts to appear. So for example, you could imagine that some metal ions, maybe lanthanides, maybe some other metals, when they are coordinated by some prebiotically plausible ligands, they may be able to have the catalytic ability to activate the trimetaphosphate for the triphosphorylation of hydroxyl groups. This is just one possibility. Of course, there is also the possibility that before those 5' triphosphates, you may have had different activation groups. And one model system that was used for a long time, or is still used in the field, is that these 5' phosphates are activated with imidazolides. And there may have been different activation groups used very early on. And I like the idea that during the RNA world, we started to get to 5' triphosphates, just because every single living organism on this planet uses nucleoside triphosphates as energy source, so that we have this continuity in the metabolism and in the genetic information. So in a sense, I cannot answer your question. We are still waiting for those miraculous catalysts to be found by researchers in the field.
Thank you.
¿Alguna otra pregunta? Tenemos tiempo para preguntas, por favor.
There has been founded any transcript from some life being that have a similarity in sequence or in structure or function, or the idea is that these possible ribozymes are no longer existing today?
So no ribozymes have been found in nature with any sequence similarity. In theory, that might be possible. It would be an astronomical coincidence that one of the sequences that was selected here in an environment that never had contact with biology ended up as the same sequence as in biology. And the other reason for that is biology doesn't have those ribozymes. Biology has the ribosome, the spliceosome, group 1, group 2 introns, and self-cleaving ribozymes, and RNase P. But it doesn't have this instrumentarium of other ribozymes that may promote a metabolism, which is mostly taken over by proteins these days. So there might have been ribozymes similar to those in the RNA world, but they don't exist anymore because their function has been taken over. I also may want to add that most of the ribozymes discovered to date would not be of use in an RNA world. For example, with these triphosphorylation ribozymes, we showed, yes, trimetaphosphate can be used as an energy source, and the chemistry can be done pretty easily. But these exact ribozymes here probably were not useful in an RNA world, because these ribozymes cannot triphosphorylate free nucleosides. They can only triphosphorylate long RNA molecules that have a 5' hydroxyl group. But we are working on those, and we are trying to, the long-term goal of my career is in the next 20 to 25 years, to make all the ribozymes necessary to have a small self-replicating RNA system, and then we watch an RNA world organism in the lab evolve.
Por favor.
Thank you. Thank you very much for your lecture. And I don't really know how this replicative ribosome works, but in the RNA world, in the mature RNA world scenario, how can the evolution of the ribosomes by mutations could have done? Because you said that there is a number of mutations that are needed for the ribosome to increase their activity and so on, but how the mutations can be incorporated into the ribosomes?
So your question is, how were mutations generated in our RNA world? So if we assume that there was a ribozyme in the RNA world, or any activity that polymerized RNA, then this activity would be most likely more error-prone than today's RNA polymerases. So each RNA polymerization in an RNA world, whatever its catalyst is, would make a number of variants. Let's say it makes 10 different copies. And many of these RNAs would have mutations. Only those copies that have the best, most efficient sequence would be able to help the replication of the cell. And it's actually more complicated than that. because you will find some sequences that then tolerate mutations better. Because in RNA world, you would expect to be more error-prone, and you would evolve towards ribozymes that can tolerate a lot of mutations. So I never claim that these in vitro selection experiments we are doing happen in the RNA world. Those are completely different things. We're just trying to show or to test, do these activities exist? And if they exist, how do they function? What is their chemistry? Once in the RNA world, that was a totally different scenario, and how the RNA world originated is probably closer to what Ram described. We had a very messy origin. We had all types of chemistries going on. There was some type of selection process that we had RNA strands or DNA strands. And there was always enough room for error. So I think it's not a problem to think how we got enough mutations. It's more the other way around. How could we have ribozymes that have few enough mutations so that we still have functional molecules?
Thank you.
Allá atrás, por favor.
A ver, en Estados Unidos, desde lo que yo calculo, 1860 y tantos, ya Estados Unidos era un consumidor importante, pero empezaba de grandes cantidades de alcohol, consumo de alcohol, marihuana, cocaína, opio, etcétera, etcétera. Estas gentes que ya llevan varias generaciones, muchas familias en Estados Unidos, que ya llevan muchas generaciones, ¿qué tanto se replica o qué tanto se transmite esa adicción por estas drogas o alcoholes? Al final de cuentas todas son adicciones. ¿Qué tanto esa réplica subsiste o qué tanto se puede experimentar tratando de plantear qué tan importante o cómo es la réplica de estas especies de adictos, repito, en Estados Unidos, porque es importante el mercado, cómo ha estado impactando en las nuevas generaciones y si hay algún indicio de si hay evidencias de esa réplica?
Well, thank you for the question. It's well outside of my expertise, so I'm sorry I'm unable to address that question. We can talk later about drugs in the United States, but it's far outside my expertise on catalytic RNAs.
Bueno, una cosa que es una realidad es que absolutamente todas las civilizaciones conocidas han encontrado algún tipo de droga, incluyendo el alcohol, algún tipo de matemáticas, algún tipo de poesía y algún tipo de música. Si esto está relacionado o no, no lo sé, pero ciertamente no se puede suponer que solamente una cultura ha sabido utilizar las drogas en cierta dirección. Hay una tradición larguísima en las etnias mexicanas, y creo que no necesito entrar en detalles, sobre todo para evitar ir a dar a la cárcel, de uso de distintos estimulantes que se clasificarían como drogas actuales.
Gracias.
Pero yo tengo una pregunta. I do have an actual technical question for you. Has anyone attempted to develop one of these catalytic ribozymes, like the one you are describing, with the different canonical bases, going beyond the canonical 4 bases that we know of?
As far as I know, it has not yet been attempted with catalytic RNAs, but Stephen Benner, I think it is his lab, who developed an artificially expanded genetic system and who did an in vitro selection with that system to find RNA aptamers, so RNA molecules that don't catalyze reactions, but they very tightly and very specifically bind small molecules. So he showed that yes, this artificially expanded genetic system is able to undergo in vitro selection for the fittest sequences. And I think it's only a matter of time that his system or a similar system would be used by people to find catalytic RNAs.
Respecto a los lantánidos, los orbitales de son muy importantes y entonces pueden hacer coordinaciones con el fosfato, pero ¿no será que son demasiado fuertes?
Yes, and that's exactly the point I try to make, that lanthanides are so strong Lewis acids that they not only are able to help here in the catalysis by coordinating the negatively charged oxygens on the trimetalphosphate, they activate this phosphorus for a nucleophilic attack by water, or in our case, by nucleophilic attack of the RNA 5' hydroxyl group. But as you see here, those RNAs that we're able to isolate by in vitro selection, when they react in the presence of these lanthanides, there's a lot of degradation going on. And this, I think, is exactly the reason that you mentioned. It's because these lanthanides are so strong Lewis acids that they can catalyze all kinds of reactions, all kinds of acid-base reactions that lead to the hydrolysis of RNA.
¿Alguna otra pregunta? Por favor.
Gracias. Good afternoon. I have a question, and it's about you mentioned that the trimetaphosphate can be used as a source of energy with the pH of 7. But how, why in that kind of pH, it's like a neutral, but what chemistry reaction happens so it can be used as an energy source?
Just to clarify that I understand the question, you are asking what reaction?
Like, why does it happen that with the 7 pH, it actually works as an energy source?
Okay, I think I can address your question with a supplemental slide, this one here. So we tested the dependence of this triphosphorylation reaction on the pH of the reaction. And here, vertically plotted, is the logarithm of the observed rate constant, and here's the pH. And what you see is under two different conditions that differ in magnesium ion concentration and trimetylphosphate concentration, you get a very nice linear correlation. And the slope in both cases is almost exactly 1. And what that tells us is, at increasing pH, the reaction, if you increase the concentration of OH- ions in the reaction, then the reaction goes by tenfold, then the reaction goes tenfold faster. And what that tells us is that the rate-limiting step in this reaction is the deprotonation of a single group. And in our case, I think it is quite logical to assume that the rate-limiting step is the deprotonation of this 5' hydroxyl group, so that you have a negatively charged oxygen, and that can make the nucleophilic attack on the phosphorus here. And there may never exist a negatively charged oxygen, but the ribozyme may push and pull a few electrons so that you have this negatively charged oxygen there. But this correlation here shows you the dependence of the catalyzed triphosphorylation reaction on the pH. And it tells you something about how the reaction works. Does that address your question?
Ok, bueno, creo que es tiempo de continuar. De cualquier manera, el profesor estará aquí con nosotros el resto de la semana para el resto de las actividades del simposio. Por favor, agradezcámosles nuevamente con un fuerte aplauso. Y vamos a continuar con la siguiente conferencia. So, the second lecture will be given by Professor Marie-Christine Morel. Professor Morel is an academic from the Faculté de Biologie at the Pierre and Marie Curie University and at the Muséum National d'Histoire Naturelle in Paris, France. The main research interests of Professor Morel's laboratory are centered in the study of small ribozymes under extreme conditions, the interactions between RNA and cell compartments, as well as the peptide-liposome interactions, and the study of the persistence and adaptability of viroids, these subcellular viruses considered as vestige of ancient life. The list of publications of Professor Morel includes not only an important number of articles in high-impact journals, but also a considerable number of books for specialized and non-specialized readers. She has co-organized and participated in several international conferences of her area of expertise. Professor Morel has supervised the research activities of PhD, graduate, and undergraduate students, and has also participated in important administrative duties in France. The title of her lecture is From Ancient to Modern Worlds, Plausible Molecular Fossils, Viroids and Ribozymes. Ready, please?
Une minute. Okay. I'm waiting 1 minute. So during this minute, I would like to thank very much the organizers. I have no micro. Yeah, thank you. I would like to thank very much the organizers of this beautiful symposium, and especially Antonio, my friend Antonio, who is a very kind scientist, kind people, and moreover, he is one of the most important ambassador and tireless worker on the origin of life all over the world. So everybody working in the origins of life must thank you very much, Antonio, for all the things you do in this field to assemble all the people and to spread our work. So I have again 1 minute talk to wait for my name. You think that? No.
Because you are in the, so go to, it's in French.
Yeah.
Fish here. Page, mise en page. And then, this is okay, this one?
No. Oh shit.
Because it's white screen, you have to change it to 4 by 3. Or maybe disconnect it and put it back again.
Because when I try...
Just wait. And now try again. Your PowerPoint presentation. Yeah, PowerPoint presentation. Yeah.
Okay.
It should work now.
No.
You have to change it to 4 by 3. Yeah, that's the 4 by 3 aspect ratio you have to change. Okay. Where is page setup? You know, a page setup page. Under the Fisher, it should be page setup. Or go to this, what is that?
No, web page.
Page setup, you know, there's a page setup. It's under the PowerPoint. Open the PowerPoint presentation.
Wait a minute. No PowerPoint.
It should be under the Fisher.
No. It's here. So,
No, you are the correct one. You are at the right one. You are at the 1024768. That's correct.
That's
No,
correct?
yeah, that's correct.
Ok, désactiver, détecter les moteurs. Sorry, wait a minute.
So you see, it's not really right. What you need to do is change the aspect ratio to 4:3 and it will work.
No.
It should be, usually it's under this page.
No.
And then you click there, click under transparent. No, click under transparent. No, yeah, that is right. That's the one that you want. So say okay.
Okay.
Just here it should change. And then go back to PowerPoint
No,
presentation.
no, doesn't.
Oh no, it is still coming out like this.
Shit, c'est pas. Because it works at before the. I don't understand what happens. Shit de merde. No. So I have a key. I can put it in this on your computer. No? Oh, yeah, okay, yes. Yeah. To confirm, to go back. What the? Non. Qu'est-ce qu'il m'a foutu là? What do you do? Attends, un moment. Yeah, okay, good. Thank you very much. No problem. Thank you very much. So, I would like also to thank Sandra for her presentation, but I was disturbed a little. So, you know everything about the RNA world, thanks to the excellent presentation yesterday and today. Of course, we have to know a little bit, but I think that you know a lot of things from yesterday and today presentation. I will talk on, although you know a lot of things, I want to place the RNA world in the biological past over 4 billion years ago. So my presentation will be start with a short introduction with the biological past over 4 billion years ago. Then I don't understand what happens with my techniques today, but no, it doesn't work too. Okay. Moment. Do you have a pointer? Please. Thank you very much. So I will start with the biological past over 4 billion years ago. Then I will present viroids and the ribozymes, including the biochemical and biophysical experiment on these viroids. Then I will talk about the adaptability of viroids and I will conclude with some evolutionary aspects. So, okay. So the leading thread to imagine the biological past over. I know. Oh merde. Oui, oui, je vais le faire de là. Okay. So the leading thread to imagine the biological past over 4 billion years ago is based on two principles. The first principle is the principle of continuity that links the predecessor with their descendants, and it is assumed that, for instance, metallic compounds may have arisen early in evolution to drive the energetic, the structural, and growth needs of primitive organisms. And as a matter of fact, for instance, iron, sulfur, magnesium, sodium within modern proteins and nucleic acids are an adaptation retained from early mineral biochemistry. So the second principle is based on the evolutive jump and exaptation. And for instance, it is well admitted that some organic cofactors, for instance, adenine, modified nucleosides, nicotinamide adenine dinucleotide, probably predate the RNA world. And as you can see here, the 2' phosphate of NADP, the 3' phosphate of coenzyme A might be the remains of ancient RNA backbones. So we know today that RNA performs genetic role, catalytic role, thanks to ribozymes and small ribozymes, essentially, that is hammerhead ribozyme, hairpin ribozyme, twister ribozyme, etc. And RNA performs structural and regulatory roles. And it is more important to underline that small RNAs travel between organisms as well as between kingdoms domains. And we know today several non-coding RNAs that act as sensors, and small non-coding RNAs such as microRNA for the well-known non-coding RNA act as regulators of gene expression. Hence, the RNA world hypothesis formulated by Gilbert in 1986, but already proposed by, as you know, Francis Crick, Carl Woese, Francis Crick, and Leslie Orgel. Leslie, who stated in 1968 that the question of catalytic activity of polynucleotides remained open 40 years ago. Hence, we know today this means that RNA came before the DNA, RNA, and protein world. So, what can we say about RNA as a primeval catalyst? We know today almost 100 modified nucleosides. And from the phylogenetic analysis of these modified nucleosides, one can assume that 15 of them were present in the last common ancestor, and from the structure and chemistry of some of them, some can could have been present in the prebiotic world and in the RNA world. And for instance, as early as in 1971, Friedman synthesized the nicotinamide in prebiotic conditions, that is from ethylene and ammonia. Hence, the ribonucleotides in cofactor and modified nucleosides could be the remains of ancient ribozymes. As you can see here, the topology of several hammerhead ribozymes found in all the domains of life, in bacteria, plants, metazoans. And just a word to remind you that the word hammerhead was given by the Foster and Simons, two Australian scientists who described the first hammerhead ribozymes. And they found that the structure resembled the mouth of the shark hammerhead frequently encountered in the Australian coast. So it is why we talk today about hammerhead ribosome. So these hammerhead ribozymes were detected as ultra-conserved motifs in the genome of amniota, reptiles, birds, mammals, including humans. And the idea is that these small extant ribozymes are living fossils of a primeval RNA world that would have remained in the genome of modern organisms. A less known competence of RNA is the result of its fragmentation as part of responses to cellular stress, temperature stress, salt stress, pressure stress, and all the canonical RNA, that is tRNA, messenger RNA, snow RNA, generate pieces of RNA, that is small RNA, that bind other transcripts, that mediate immune stimulation, that changes a repressor into activator, and conversely, etc. Thus, RNA pieces perform functions distinct from their parents, and it is a kind of exaptation, and this underlines the fact that RNA is at the origin of several evolutionary innovations. We know today that tRNA is considered as ancient molecules that serve several biological functions. And as you can see here, from the cruciform structure of tRNA and from their fragment, that the structure resemble the tRNA like at the end of RNA virus genomes. And as you can see here, the cruciform tRNA-like motif are also found in the citrus exocortis viroids and in the chrysanthemum stunt viroids at one end of the rod-like structure. So this slide allows me to introduce the viroids. So viroids are discovered in 1971 by Theodore Diener, who want to know what happens with plant disease because of an agent that causes this serious disease in potato. So as he observed twisted bracts and wrinkled leaf surfaces of potato plants, tubers elongated with pointed ends, and eyes deep and prominent. So he asked the question, is it possible to characterize the nature of this agent that causes this disease from leaf extract? So he performed a series of experiments, and for instance, after prolonged ultracentrifugation, he noticed that the infectious principle remained in the supernatant. The treatment of extract with phenol did not lead to changes of the sedimentation properties, and among other experiments, he was able to suggest that the infectious material was a free nucleic acid and not a conventional viral nucleoprotein particles. And furthermore, the incubation with ribonuclease A but not incubation with deoxyribonuclease, abolished infectivity. Thus, he concluded that the infectious agent from infected leaves was free RNA, and he named these free RNA viroids. So one can observe here, magnified by electron microscopy, the relative sizes of DNA, the T7 DNA from the bacteriophage T7, as compared to the size of PSTVD, that is the potato spindle tuber viroid. And you can see here, also magnified by electron microscopy, the linear and the circular strand of this viroid. So viroids are naked small RNA. Some cause devastating plant diseases, others are asymptomatic, and others are beneficial. So it is very difficult to find viroids. The symptoms are related to the environment. For instance, one observed that the circadian temperature variations impact the replication cycle. And some possess ribozyme motif. It is why I was interested in viroids initially, because some possess ribozyme motifs, and almost 30 species were so far identified. So you can see here some pathogenic effect on plants, on coconut kadang-kadang, in avocado, on apple, on peach, on chrysanthemum, on grapevine, on citrus, on tomato, on potato, and on Mexican pepita. So it is very interesting because Mexican pepita has been isolated from plants growing wild in the Mexican state of Aguascalientes. And MPVD nucleotide sequence is closely related to those of the tomato and those of the potato and other viroids. And the phylogenetic and ecological data indicate that MPVD is the ancestor of tomato viroids, potato viroid, and other viroids. So the ancestor came from Mexico. And the hypothesis formulated by Martinez, Soriano, and Diener, when he was a postdoc in Diener laboratory. So the hypothesis is that potatoes grown in the United States have become infected by chance transfer of MPVD imported from Mexico as germplasm in the 19th century, when Mexican wild species were introduced to obtain mildew resistance. So I don't say anything else. So regarding the viroid trafficking, as opposed to viruses, there is no viroid receptors, so the viroid enters the cells through the pores in the cell membrane, through mechanical breaks that occur by tools, wind, via insects, etc. Viroid may be latent for several years, and they can move intracellularly, as you can see in this picture, intercellularly, as you can see here, through plasmodesmata, and to distal part of the plants through the phloem, that is the vascular system. So this is the trafficking of the viroids. How they enter and exit the nucleus and the chloroplast is today completely unknown. So I said that we know almost 30 species. And on the basis of sequence homologies of all known viroids, and on the basis of the self-cleavage capacity, viroids have been classified in two families. The first family is the family of Pospiviroideae, including 30 species with a rod-like secondary structure. They replicate in the nucleus of the cell, and they possess a central conserved region named CCR. The most representative of this family is the potato spindle tuber viroid, PSTVD, and the Mexican papita viroid, MPVD. The second family is named Aphson viroideae. It includes 4 species with rod-like or branched chains, and they replicate in the chloroplast of the cell, and all members of this family possess hammerhead ribozymes. And the most representative of this family are avocado assemblage viroid and chrysanthemum chlorotic mottle viroid. So you can see here the secondary structure of that is the rod-like secondary structure proposed for PSTVD, that is the representative of the Pospiviroideae. In B, the quasi-rod-like structure proposed for the avocado assemblage viroids, the type member of the Aphson viroideae, and in C, the complex branched structure proposed for the chrysanthemum viroid, a member of the Aphson viroideae family. So the primary and secondary structure of Pospiviroideae are represented here. It is divided in 5 structural and functional domains. The first one is the CCR, the central conserved domain, the pathogenic domain, the variable domain, and the terminal right and terminal left domain. And it is the specific secondary structures of CCR that play a very important role in the replication cycle of the viroid. Interestingly, the citrus exorcortis viroid, that is a Pospiviroideae, possess a putative self-cleavage site at the bottom strand of the CCR region with a hairpin structure, but whether cleavage at this site plays a role in the replication is so far not clear. Regarding the second family, this family lacks CCR, but possesses a self-cleaving hammerhead ribozyme. So here is represented the avocado assemblage viroid, that is the smallest viroid of this family with a rod-like structure and an hammerhead ribozyme, here, represented here. And the largest viroid of this family is the chrysanthemum chlorotic mottle viroids with a branched secondary structure, which is represented here, and this secondary structure folded to give rise to the hammerhead ribozyme of 67 nucleotides. Here you can see the minimal structure of the hammerhead ribozyme with the substrate strand in blue, the enzyme strand in red, and here the cleavage site in green. So the viroid replicate according to a rolling circle mechanism. For the Pospiviroideae, it is an asymmetric rolling circle mechanism. That is to say, the infectious circular plus RNA is copied continuously by an host RNA polymerase II into a concatenameric minus strand, which is cleaved here, producing a monomer that is copied in a concatenameric plus strand, which is cleaved here, leading to a monomeric strand, then circularized to produce the circular plus progeny. The second family, that is the Avson-Viroidae family, replicate according to the symmetric rolling circle replication. This symmetric rolling circle proceed like this. That is, the infectious circular plus strand is copied continuously, giving, thanks to a nuclear encoded polymerase, that is an RNA polymerase from the host, and it is copied so in a concatenameric strand, producing in blue these strands, which fold to give rise to the ribozymes. Then the ribozyme cleaves the strands to give the monomers, which is circularized and which is copied again in a plus strand in red, which fold to give rise to the ribozymes, producing the monomer, which is circularized to yield the circular plus progeny. So 3 steps: an RNA-RNA replication with an host RNA-dependent RNA polymerase, the cleavage thanks to the hammerhead ribozyme, then the circularization by ligation thanks to an host ligase, or possibly by self-ligation. You can see here the mechanistic aspect of the cleavage reaction, that is to say, a trans-esterification reaction. The initiation proceed via nucleophilic attack on the 2' by the 2' hydroxyl group to the adjacent phosphorus, leading to the transition state. Then the departure of the 5' oxy anion leaves the 2', 3' cyclic phosphate. So the reaction can be accelerated by a general acid-base catalysis, or, and we will see, by conformational effects facilitating the formation of the transition states. And it is interesting to notice that the ligation reaction is the exact reverse of the cleavage reaction. So we decided to study the cleavage reaction of the avocado assemblage viroid, and one observed on this kinetic study that the avocado assemblage viroid plus, of the plus polarity, is less efficient than the avocado assemblage viroid of the minus polarity. So we decided to pursue this investigation, and one observed on native gel electrophoresis that several conformations exist on the plus polarity of the avocado assemblage viroid. This is confirmed by temperature gradient gel electrophoresis. One observed one major profile for all temperature for the minus strand of the avocado assemblage viroid, whereas the avocado assemblage viroid plus adopt alternative folds at lower temperature. So we pursued with a shape method, that is a selective hydroxylacylation analyzed by primer extension, and we when obtained the secondary structure by this method, and moreover, we showed that there exists kissing loop interaction within the hammerhead of the minus strand, which is represented in the blue line, that may play a role in vivo replication cycle. So we decided to go further in the investigation of the role of conformation of in the replication cycle of the viride. And for that, we used a biophysical method that is small angle neutron scattering that allows us to obtain the molar mass of the monomer, of the dimer, of the concatenator, of the particles, of the mixture, and that allow us to obtain the radius of gyration radius of the particle. That is a measure of the conformational structure. And one observed by this SANS method, that is small angle neutron scattering, the temperature dependence of the dissociation between the monomer and the dimer. And as you can see here, the scattering intensity values show the temperature-driven oscillatory behavior in the avocado assemblage viroid minus polarity, as well as in the hammerhead ribozyme minus polarity. And this temperature-driven oscillatory behavior shows that the dimer appears at low temperature, then the monomer, then the dimer, then the monomer, then the dimer, etc. So we decided to trace the Arrhenius plot of these structural results with, and one observed that the dimer dissociation in this curve is, as a function of temperature, parallels the catalytic data with a break at 27 degrees. So the interpretation, and you have here the activation, the energy of activation at low temperature and the energy of activation at high temperature. The energy of activation is calculated from the slopes of the curve. So the interpretation is that to reach the transition state, more energy is needed for the dimer at low temperature than for monomer at high temperature. So it is why dimers are less active than monomers. So now we try to understand how, not why, but how. So again, sorry. So to understand how this happens, we proceed through modelization. And as you can see here, in A, the secondary structure of the monomers showing intramolecular interaction between the H2 loop with the H1 loop when the monomer folds as it appears in this modelization. And furthermore, there are, when two monomers are end-to-end and not side-by-side, what is shown by the radius of gyration, when two monomers are associate, the radius of gyration showed that they are end-to-end and not side-by-side. So when two monomers are end-to-end, there are intermolecular interaction between the 3' ends with the H2 loop of the second monomers. So you can see here the three-dimensional representation of the monomers, and here the three-dimensional modelization of the dimer. In black, the first monomer. In gray, the second monomer, and the interface between the two monomers is zoomed here, and it explains why, how how the dimer is less active than monomer, because when the second monomer is linked to the first monomer, the interaction of the 3' end of the first monomer with this loop forbid completely the folding of this monomer, that is the interaction between the H2 loop with the intermediary H1 loop. So it is possible to understand the why and how question in biochemistry and in biophysics. So the first conclusion of this part of this presentation is that viroids are small naked RNA. They do not code for any protein. Some possess catalytic properties through ribozymes. They are rigid and flexible. There exist two non-symmetrical polarities, plus and minus, with different structure and different activities. Each polarity could play a distinct role during the viroid life cycle, and the structural motifs serve as promoter, recognition site, etc. during the processing. Now, the big question is whether viroids can replicate in other types of plastids and in other species than plants. This remains to be demonstrated. So the background, the arguments are that we know two locations in the cell for viroids, that is the nucleus and chloroplast. These argue in favor of adaptability to different environments. This is plausible. We know that viroids are at the same time rigid and flexible. This enhances the survival probability. So we decided to study the adaptation to different hosts. In the first set of experiments, I will show our result with non-photosynthetic eukarya, that is the replication of viroids in Saccharomyces cerevisiae, and then the replication of viroids in a bacteria, and in particular in the cyanobacteria. So the replication of avocado assemblage viroid in the yeast Saccharomyces cerevisiae proceed like we have already seen by the symmetric rolling circle mechanism. And we constructed this vector, allowing the constitutive expression of avocado assemblage viroid in yeast. And one observed by northern blot that the extract of yeast infected here by plus polarity, when this extract is loaded on the gel, incubated with riboprobe plus, one observed replicated RNA minus strain, minus polarity. And when the extract of yeast infected by minus polarity is loaded on the gel and the gel incubated with riboprobe minus, one observed the replicated RNA plus. So one observed through the series of experiments the transcription, replication in the yeast, the efficient cleavage by the hammerhead ribozyme, and the circularization. So we also studied the persistence of the viroid in Saccharomyces cerevisiae, and one observed that the viroid replication occurs after 25 generations. So we showed the functional diversity and the unsuspected adaptability to a non-plant host, and this encouraged us to ask the question, is it possible that viroid replicate in another domain of life. So we investigated the replication of viroid in cyanobacteria. I remind you that chloroplasts found in plants have their ancestry in cyanobacteria via a process called endosymbiosis. And we tested this replication cycle already. I already talked about this. That is, the plasmid, we constructed the vector, and the plasmid is transcribed in the cyanobacteria. Then one followed the RNA replication, then the self-cleavage and the ligation. The cyanobacteria, for cyanobacteria, we choose Anabaena, that is a filamentous cyanobacteria, that is a photosynthetic prokaryote, blue-green algae. And one observed in line 5 the linear replicate plus, which is detected by riboprobe minus when the minus plasmid was loaded, and conversely, one observes the replicate minus strand when riboprobe plus is detected, is incubated in the gel. So, the second conclusion is avocado assemblage viroid replicates and persists in Saccharomyces cerevisiae, that is a non-photosynthetic eukarya. Avocado assemblage viroid that normally replicates in the chloroplast of avocado is able to replicate in the cyanobacteria Anabaena. We showed for the first time that a prokaryotic cell possesses all the machinery required to sustain RNA replication without DNA intermediate. Hence the question, are extant viroids descendants of free-living proviroids that have invaded ancient cyanobacteria? Some in this frame, some of these organisms later became endosymbiont of primitive plants and evolved into chloroplasts, usurping the machinery of their host. However, RNA cleavage can be achieved by hammerhead ribozymes in the Afson viroidae, and the polymerase and ligase activity might have been lost during, by the viroid ancestor during evolution, since they have this polymerase and ligase activity have been discovered by the selects presented by Uli in artificially evolved RNA. You can see here the PDB, the three-dimensional PDB of the RNA polymerase ribozyme found by Jerry Joyce, and the RNA ligase ribozyme first found by Bartel. So, a few words to conclude with the evolutionary aspect. We know that viroids and related species, such as satellite RNA, hepatitis delta virus, are distant from viruses. There is no link. So, one observed that these putative free-living fossils of the precellular RNA world share some properties, that is small size, the circularity, the replication through a rolling circle mechanism, and ribosomic activity, meaning that an RNA genome performs by itself its own, is capable to perform by itself its own replication. So my last word is that the viroid first hypothesis must be, of course, further experimentally and phylogenetically investigated. This is one of many exciting avenues, a kind of El Dorado, a a very promising land for the young scientists, for the students, not especially because viroids originated in Mexico and that the ancestor of all viroids are probably coming from Mexico, but because it is very, I think that these species are very important in order to go deeply in the RNA world studies and in the origins of life understanding. I thank you very much for your attention. Gracias por su atención. Okay, thank you. Okay.
Thank you very much, Marie-Christine, for this marvelous and moving, touching conference. Tenemos tiempo para preguntas, por favor. Recuerden que pueden formularlas en español y en la traducción nos ayudan a proponerlas en inglés. Por favor.
It's just a very simple question. I remember to have heard about viroids back in the 80s. So my question is, has it been very difficult to study viroids? Because it seems to me that you are just starting to study them after all these years. Has it been very difficult?
Yeah.
And why?
Yeah, it's a very good question. It is why I encourage you and all the students to go deeply in the knowledge of viroids, because there are very few laboratories working with viroids. I don't understand why. Maybe, I don't understand. My bad interpretation is that, unfortunately, biologists are very less interested than chemists in the study of the RNA world and in the study of the origins of life. I am right. In France, in Europe, it is the case. So, and viroid is a biological species. It is not a chemical species. And I am afraid that the people, the researchers that may be interested in go deeply in the RNA world negligate completely this species. And it is a pity. So there is an Eldorado for all of you that want to go, that all of biologists that want to go deeply in the RNA world and as a biologist. You know to be a biochemist. Yes. You must be a biochemist, but biology is good.
So the transfer across species lines is extremely impressive. So are we in this room possibly infected with viroids? And what is the most sensitive experiment to detect them?
Yeah. So as I said, some viroids are asymptomatic and asymptomatic. So, and some viroids are beneficial. As you, in Brazil, some cultivate lemon plantation with infected by viroids in order to maintain the tree dwarf. This is the word, small, and this facilitates the harvest of the record of the lemon, and this is an economical benefit incredible. So some are beneficial according to the economic field, but some are also beneficial according to the production of the grape, in wine grape. And this was demonstrated in Tunisia. Of course, country which are dependent of agriculture culture are very interested in viroids, and they try several. But how can we detect viroid in this room? I don't understand. I don't know. I don't know. Maybe when. No, maybe I don't know.
Por favor. Hay otra pregunta.
Very interesting. Can you tell us what are the bases for the pathogenicity when they are actually a pathogen? And if resistance exists, what is the resistance based on?
I don't know any resistance to viroids. And the basis of pathogenicity, the basis is the rate of replication cycle. And one observes pathogenicity by observation of the leaves, of the fruit, of the fruit. So what do you mean by basis of pathogenicity?
It seems, at least to me, unlikely that just through the sequestering of RNA polymerase II function, you would actually manage to generate, for example, the deformation in the potato, no? Yeah. So I was wondering whether there was something else, whether either the activity of the molecule or interaction with other things increased the pathogenicity of the viroid.
No. To my knowledge, there is nothing to do.
So how do the infections proceed? So if you infect an orchard of avocado, will it essentially eliminate the trees, or what's the evolution of an infection by virus?
The evolution of the infection is the devastating the avocado plantation. So there is no medicine against viroids as far as I know.
Christine?
Yeah.
Nice talk. Are you planning to infect archaea to try to mimic the nuclear viroids? I mean, the origin?
Yeah. We tried, but it fails, but not for scientific reason. Because one of the collaborators was in Japan, I was in France. It was very complicated. But we tried, and we have the vector. If you want to try, I can give you the vector. It is construct, and but it is of course we need to try with the archaea bacteria.
Yeah, perhaps not me, but some collaborators in Spain, as Ricardo Flores, that you know.
Oh yes, of course. But Ricardo Flores,
perhaps
we will talk about
eventually.
Yeah. Of course, I know very well. And Ricardo Flores give us all the initial plasmid for doing our work.
Thank you.
¿Alguna otra pregunta?
No? Oh, sorry.
Allá atrás y aquí adelante, por favor. Y acá adelante también.
So, can I ask a question? Okay.
Never mind. It's okay.
Do you think there could be like an RNAi type medicine? I mean, could you target these with sort of an RNAi type medicine where you target them with an RNA degrading system, where you put in a 20-tumor RNA complement to try to have it degraded by a risk complex, say?
Yeah. So you proposed.
I'm proposing a medicine.
Yeah, I understand. Yeah, maybe. But you have to enter this. Yeah, it's possible, of course. Mm-hmm. But I must say that viroid is not my field of research. My field is ribozymes. I was interested in viroid because of ribozymes and because I am a biologist. And I talked today, I developed this viroid in detail because Antonio said, you must explain what is a viroid to these young students. So, but I am sure that no medicine exists. And I am sure also that few laboratories are working in this domain. It is a pity. So, Pepe?
Thank you very much for your talk. Is it well understood the mechanism of infection of these viroids in plants? I mean, I suppose there is a kind of RNA interference. If it is that so, what it could be very specific mechanisms. I mean, some genes, plant genes are affected specifically.
Yes, it is possible to cut viroid by RNA interference. Yes, I have not tried in my lab, but I think it is possible. Sorry, but I am not really competent in this.
Si le acercan un micrófono, por favor, a la persona de atrás.
You say that the virus might lose their polymerase and ligase activities, but why do they, these viruses prefer infecting cells than their free living style? I don't know.
I do not understand very well the question. Why viroids infected?
Why do they prefer infecting cells and continue with their free living lives?
Antonio, qu'est-ce qu'elle dit?
Probably about the pathogenicity. Why do they infect instead of just living in the host cell?
What?
Why do they infect the host cell instead of just living inside? Why do they prefer to attack?
The infection is that they live inside.
Okay.
They live inside, so
es que usted comentó que los virus perdieron su actividad de polimerasa y ligasa.
Okay.
¿Puedes repetir, por favor, en español?
Sí. Usted comentó que los virus perdieron su actividad de polimerasa y ligasa y prefirieron infectar las eucariotas. Bueno, en vez de continuar con su vida libre. ¿Por qué?
Yeah, but maybe the majority of viroids prefer the freestyle living. Who knows? So maybe here, the freestyle living here. But I said that they lost polymerase and ligase activity during evolution, and because they used the polymerase and the ligase of the host. So if we retain the hypothesis that viroids are free-living organisms on the ancestry of the first RNA world, so we must conceive that before they possess polymerase activity, ligase activity, cleavage activity, etc. And if we, in this frame, they have lost these two activity and they usurp this activity from the host. That's all. This is an hypothesis, this is a speculation. But this speculation is sustained by the fact that some viroids possess ribosemic activity, and there exist, thanks to the SELEX method, that is the in vitro selection, some RNA capable to be polymerase or to be the ligase. So nothing is impossible with RNA.
Por favor.
You said you're not an expert on viroids, but I'm sure that most of the people in the audience had never heard about viroids, which I think is something to be kept in mind. And secondly, given the phylogenetic vicinity of fungi and animals, Have you tried to infect animal cells with viruses?
No, only Saccharomyces cerevisiae and cyanobacteria. And as I know, nobody tried to infect animal cells. Bacteria was tried several times, and I don't know why it failed, and we succeeded in our lab. Thank you. And but nobody tried to infect animals or fungi or other organisms. So you must try.
No, I prefer to keep away from pathogens.
Yeah.
But I have another question. The RNA polymerase II, which is employed during the replication of viroids, is also the same one used for the replication of viruses.
No, there are a factor associated, a cofactor associated with RNA polymerase II. And this factor is correctly characterized by Ricardo Flores, yes.
And no one has ever attempted to use monomeric RNA viral polymerases?
What?
Like if you have RNA viruses, they encode for a single unit polymerase. Has anybody attempted to use them? So it's an open field, really?
Yes, it's an Eldorado, I say.
Por favor.
Excuse my ignorance, but the fact that you get the hammerhead ribozyme coated by the two strands by the positive and the negative strand, is this something that is seen in other places, or that's not my question? This is just
is something that
is this something that is seen in other situations? The fact that you get an active hammerhead ribozyme coated by the positive and the negative strand.
What is the question?
Well, look, actually, there's a second question to it, because I assume that this means that there's very strong constraints to the changes in the sequence, because if you can make a hammerhead ribozyme from the positive and the negative, then the amount of positions that are invariable must increase, no? Have you managed to check this?
No, I have not. No, but I agree with you. There is a sequence constraint, very important, between the two polarities.
Por favor, there is another question over here, please, Mary Christine.
I was struck by the examples that you showed first where the viroids were found. You showed all plants, all plants. And then you started wondering whether they can infect cyanobacteria. And then you put them into cyanobacteria and showed, yes, the same thing can happen. My question is this. Given the fact that viroids have been detected only in highly evolved species and not in the really primordial ones, could it be that the viroids are much later? In terms of their, they were products of much later evolved life, that they could have infected only the highly evolved species, because you don't find them in the very primordial species that you would expect if this was present at the origins of life?
Question, you said that from the picture of the infection of several plants, this and not.
And then you extrapolated it back to the bacteria. But so far it has not been found in bacteria naturally.
Yeah.
So the question is, could the viroids have come much later because they are only present in evolved species?
Why? No. The fact is that we detected viroids due to their pathogenicity. And the pathogenicity is flagrant on agriculture, and it is why Diener tried to characterize the agent of this pathogenicity. But like medicine for human, you can have a disease, but if there is no symptom of this disease, nobody want to characterize your disease, which is not a disease because it is asymptomatic. You know? So it is the same reasoning for all the domains of research in medicine or in agriculture, etc. So, and there is no link between the fact that they were detected in higher plants and not in bacteria. I think that there is no link. Maybe there is one, but I think that why, in your reasoning, why you could infer that there is a link?
No, because it goes back to the hypothesis that they had ligase activity, they had polymerase activity, but then they lost it.
Yeah.
You know, the same hypothesis could be turned around and said, probably they didn't exist at that time and they were made much later.
Yeah.
You know, what I'm saying is I'm just playing on the hypothesis that if you could extrapolate backwards, you can extrapolate forwards as well.
No, I disagree because some years ago, ribozyme was considered as an exception, you know, and hammerhead ribozyme was some motifs, very exceptional, etc. And now we found hammerhead ribozyme everywhere in our genome, etc. So there is no link between the fact. You understand what I mean? Yeah.
The devil's advocate trying to reverse it the other way, rather than why try to put a property on the viroid which probably it never had.
Yeah, of course. Mm. But
you mentioned.
Sí, aquí adelante y vamos después allá, por favor. Por favor.
You mentioned that can be considered like a fossils, earlier fossils, but this means that it should be more spread and not only attack plants, or they came very late in the evolution.
That's the same issue. Yes, it was the same question. But yes, of course, there are a lot of questions, and tant mieux. Good, it's good. And this question is due to the fact that our field is very speculative. Of course, you know, everybody knows that origin of life is a very speculative field. It doesn't mean that it is not a rich field, but it is very speculative. So we must envisage that something was lost during evolution and something may be recovered during evolution, etc. And there is no direct link, maybe.
Por favor, tenemos una
Lo voy a
pregunta.
hacer en español.
Por favor.
Para infectar un viroide, una célula, pues evidentemente tiene que pasar la membrana. Quizá el hecho de que no se encuentre en una arquea es porque la composición de su membrana es completamente diferente al del resto de las células.
Very good remark, but we have never tried. So we will try, and it may be that this is a good reason, of course. Yeah.
Y quizá la forma.
The idea is that viroid enters the cell through a break in the membrane, you know. But maybe the archaea suffer also with a breaking membrane, although the membrane is not the same as in bacteria.
Y bueno, me imagino que quizá la forma de saberlo es sencilla si se prueba solo liposomas con lípidos pues como de células de mamíferos u otras normales y liposomas de arqueas. Y si no puede entrar un liposoma de arquea, pues es la membrana la razón.
yeah, we need to try.
Going back to the issue of the biological distribution of viroids, I think, let me play the devil advocate of the devil advocate argument. On the one hand, we pay little, if any, attention to epidemics outside animals and plants that are significant for us. And when we look at the plant kingdom, we mostly use what the people used to call the higher plants. And I have never seen any report about epidemics among bryophytes or among mosses. Who knows? We simply don't pay attention. So that could be an argument in favor of the idea that perhaps they have persisted since ancient times, going through the history of biology. On the other hand, it's equally true that plants make the most extensive use of RNA. In the sense that, for instance, the highest diversity of RNA viruses are found in plants, and the highest and most sophisticated methods of regulation via microRNAs are found in plants. So I think that the jury is out in still. I think that it's not easy. We clearly need to know more. But one thing that I will certainly look for will be viroids in bryophytes or lower plants, and in algae like ulva, and those plants that are clearly ancestors of plants. That will be a very good way of proving. Of course, you can always say, okay, there was a lateral transfer of viruses, whatever, but that will be a very good experiment to do. And you have all the technology to do it.
Yes, we will try.
Tenemos una pregunta aquí, por favor.
¿Se ha detectado alguna respuesta inmunológica de alguno de estas plantas? ¿O algún mecanismo de defensa para los viroides?
No, unfortunately, I don't know, but I am sure that nothing has been discovered so far. But we must check, because I am not sure of all my bibliography on this theme, but we have to. If you are interested, you must check. I am very happy if you are interested to
Una
know
última
more,
pregunta, perdón.
but I am sure that not.
Una última pregunta, por favor.
I hope this is not a foolish question, or maybe you've mentioned it and I missed it, but being free RNA and there being so many RNases in the environment, how do viroids survive not being degraded by these RNases? Does this have to be something to be with their structure?
Maybe the structure is very important in viroids. As I said, they are flexible and rigid at the same time. So I guess that they can adapt in several environments, and that's all what I can say. This is an important question to study too.
Ok, agradezcamos por favor nuevamente a la profesora Christine Morel. Thank you very much. Con esto concluimos las actividades del día de hoy. Agradezco a todos su atención a las actividades de esta tarde, a nuestros conferencistas, por supuesto, Ulrich y María Christine, a los coordinadores del simposio El mundo del RNA pasado y presente por la oportunidad de ser partícipe de este evento, Y les recuerdo que las actividades continúan. Mañana tendremos la conferencia Una visión estructural del mundo del RNA, impartida por Alfredo Torres Larios, así como la conferencia Los límites funcionales del código genético y cómo lo supera el RNA, por Luis Rivas de Puplana. Entonces los esperamos y recuerden que el evento continúa a lo largo de la semana. Gracias, buenas noches.